This is Joanne Corey at the Salk Institute for Biological Studies in La Jolla, California, and I am here today in my role as guest editor for the annual review of plant biology. It's a great honor for me to have this conversation with Professor Marc Van Montagu from the Department of Plant Biotechnology and Genetics and the Institute of Plant Biotechnology for Developing Countries at Ghent University. Marc is talking with me by Skype from Belgium. Together with his colleague, Jeff Shell, Marc Van Montagu discovered the gene transfer mechanisms between agrobacterium and plants, which turned agrobacterium into an efficient delivery system for genetic engineering. This enabled scientists in the field of botany and brought the study of plants into the era of modern plant molecular biology. So good afternoon, Marc. It's good to see your face.
Hello. It's a long time that we saw each other, really.
Yes. Let me start by saying to you that you're one of my scientific heroes, so I'm absolutely delighted to have this time to hear from you directly about the
I will change
of the modern
color.
plant biology field. And you're from a working class family, and I wanted to ask you, how did your family influence your scientific life? Or did they?
Well, they did it by just not interfering, by thinking that if instead of playing on the streets with the children, I was in a corner with a book and whatever it was that I was reading, that was good, and that was all. And and always when I thought that after 12 I wanted to go to secondary school, no, why not? And then at the end of secondary school, that when there they say that maybe I should go to university, that they did the effort really to make it possible that I went to university. Well, it was cheap, so it was possible. And in Ghent, the inscription fees, in money of those times, that was maybe some $100 or so a year. So that was not a...
Yeah, it's what the University of California used to be, but we're not that anymore, unfortunately. So maybe you can tell us a little bit about how you ended up studying chemistry and then moving to agrobacterium.
Oh, but that's a long way, because already in the high school I was fascinated by chemistry, and so I was reading Aerosmith and other books already, and I thought that I wanted to have a laboratory, but I started in the attic when I was at high school. So logically, chemistry, then what I found intriguing was organic chemistry, and from organic chemistry you hear that there is something like biochemistry or chemistry of the living organisms, and that's how it started.
But that wasn't a field of study when you were studying it, right? Biochemistry?
Well, not really. In Belgium, there were some interesting people, like Saint Georgi was in Liège because he had to run away from fascism in Hungary. Levi Montalcini had to run away from Mussolini. So there was some, mostly at Liège, but also in Ghent. There was a beginning at Ghent University. Teaching of biochemistry started really during in '39, and then mostly after the war, but was a lot linked with what the biochemistry in Liège was, where I said Sven Georgi, Levi Montalcini were there. In Brussels we had Brachet at the ULB, and Brachet for cell biology with Chantraine. That was international level, very high level, because actually they, for the first, broke the story that RNA was for plants and DNA was for animals.
Yes,
they showed that was our basic. So the teachers I had, the professors, all were trained by Brachet or were influenced by Brachet or this group. So there was a good atmosphere for cell biology. And later on in Belgium, with the Duve, cell biology went on, and Claude, but 2 Nobel Prize winners, went on rather well. So that indeed was a good base. And then that made that afterwards we started immediately in the world that will be called molecular biology. That was then, and the undergraduate was finished in 55 and then was still working on the structure of RNA because it was not known if it was a 2,5 or a 3,5 link in RNA. And that was interesting. It was done, well, we'll not go into details, but with physicochemical methods because there was ways, they call it dilatometry. So if an enzymatic reaction goes on and there are charged molecules that are formed, then you can have very small volume reaction. So in a thermostatic bath, you can have then, if you keep the enzyme with the substrate, then you can see the kinetics of the reaction. That was the beginning of life. And then we went to RNA phages with Walter Fiers because we were students in the same period. Jeff was some years younger, Jeff Shell, and he was in biology, and he did microbiology and taxonomy of microorganisms. But Fiers and I were interested in the phages, in the phage world, and in the late 50s, we had quite some visitors who talked to us about a certain Luria and Delbruck had done and what was happened in Cold Spring Harbor, phage world. So we were really, we had nobody to teach us that. And then Walter Fiers went to Caltech, to Sinsheimer, and he was the first one to prove circularity of Phi-X174. So that was, and so immediately when he came back, we started together on RNA phages, and it was the beginning of the genetic code. Then we started also synthesizing the triplets, but competition of Corana was a bit hard. It was too good.
He was still like that when I was a postdoc.
Yeah.
He would have his lab meetings on Saturday morning at 8 in the morning and expect his whole lab to show up. So I think he was just very intense, right?
Absolutely. Okay, and then at the end of the 60s, Walter Fiers had decided that he wanted to start sequencing RNA and DNA, but all with capillary columns and with gel methods and eluting. It was but he succeeded, and we ourselves, we thought, well, let's go ask some biological questions, and since everybody was around the late 60s switching from prokaryotes to eukaryotes, so we thought, why not studying why a bacteria is able to induce tumors on plants? But with the idea that, well, cancer being cancer, cell proliferation, a cell cycle will be always the same, so maybe what we find in plants can be useful. We really believed it. We told it to granting agencies, and they believed it, luckily. So that allowed Jeff and I to start, and we said immediately that we would fuse in our labs, because it's the result that counts and not the persons and that remained the spirit of sharing everything, and then trying, just because we felt extremely isolated. We were fascinated by the States, the quality of the graduate schools. So I agree, my first period my first trips in the States, that was to Watson's lab at Harvard, where all the people were who found RNA polymerase, the promoters, all the basic things of molecular biology were done there. So we felt very, very tiny. And we thought also that it was the system of all these seminars, good graduate schools that the top people were immediately teaching. And so we have no courses in graduate school. You just read. The libraries are there. So it's printed there. So you should know it.
So the agrobacterium story in the early 80s and late 70s involved several labs working at high speed to uncover the mechanism of agro-transformation of the plant. So I think competition can be really motivating. It can move a field at a fast pace, and it corrects errors and misinterpretations quickly. And how frequent were the new discoveries coming out of the groups?
Yes,
We're all working on agro. There was the group in Seattle, St. Louis, you guys.
Yeah. Okay. So in the beginning, when we started, we were convinced that it was an extra chromosomal factor. So it could be a virus, because we were always working with phages. Apart from RNA phages, we both had worked on phage lambda, so lysogeny, phage P1, so we knew about genomes that also can function as plasmids. We followed the antibiotic resistance plasmids, so we were absolutely studying all what is as extra chromosomal information in microorganisms. And so then try to find what is the approach. And indeed, in the beginning, well, the idea of Jeff was we absolutely have to follow what the others did, because otherwise it's too much to start from zero all alone. So we took contact with Leiden, because Skilperot had published a lot already in the 60s. Well, it was a bit adventurous what he had published, but okay, so that was the contact with Leiden. And Jeff thought, we don't know anything about plants. Well, probably we still know very few about plants, but so surely we will not grow. And in Holland they are specialists in plants, so we have to do that with Leiden. So we were working together with them, but at home we were then looking if it is an extra chromosomal DNA, maybe we can purify it. And if it is a plasmid, well, it should be supercoiled, so just with ultracentrifugation, ethidium bromide, because it will work, because Vinograd at the end of the 60s had started that, so that's what we wanted to do. But it had to be done very carefully, because it's turned out to be a 200,000 base pair plasmid, and most of these other plasmids are just small circles of DNA. And as soon as you have a nick during the preparation, of course you have an open circle and you cannot show it anymore. And there was a faint band, and in the 60s I had learned electron microscopy and Kleinschmidt spreading. I had gone to Geneva, where Werner Arber was, but there were also a lot of specialists in electron microscopy there. So I learned these techniques, I used it in phage lambda, so we then immediately started and could see the circle. In Seattle, the person who was doing there most of the plasmid studies, Stan Falco, who is, I think he's still at Stanford at the moment. So he was there doing, and people from Seattle, Gene Nestor and Gordon, went to Leiden instead of coming to Ghent to hear how sure it is that they have this plasmid. And they send then, they give phone calls, because it's a story Stan Falkov told me, to Stan Falkov. They have this plasmid, you should be able to see it also. But they never told him that it was 200,000 base pair plasmid. So Stan Falkov didn't find it. So that's the kind of rivalry that made that we had a good start and could have the Journal Molecular Biology publication. Also there we were lucky, because when we sent in the paper, nobody wanted to referee it, because they don't understand why it would be important. And so it ended up on the desk of the editor-in-chief, Sidney Brenner. And Sidney Brenner thought it was interesting, so he decided to publish it. In this first paper, we just said that the strains that are non-oncogenic do not have a plasmid. The ones that are oncogenic have the plasmid. But of course that is not very much. Today that was nobody would consider that a serious publication. But in the early 70s, plasmids were new, and big plasmids were new, and Sidney found it intriguing. Later on, now we know that there is on top even a megaplasmid of 500,000 base pairs. That is always there. So luckily we missed it because
Okay.
otherwise we wouldn't have ever written it or so. But that's a beginning. And at that moment, a postdoc in Jean Nestor's and Gordon's lab, Maridel Chilton, got very intrigued, and she was really the driving force because she was very well organized, very hardworking. So we couldn't rest, we had to go on. But luckily we were the first ones to show that you could cure for the plasmid. And that also was we found an obscure publication from a Russian scientist that if you have this bacteria incubated at minus 25 or minus 40, and then you thaw and you do that several times, then the bacteria become so damaged that they lose the plasmid. And indeed it happened. We call it the Siberian method. And that was the way of curing, because we don't have the classical ways of with antibiotics and or other. And then for transferring, reintroducing the plasmid, we were lucky that we had the visit from Alan Kerr, and that we had contact with Alan Kerr in Australia, because he had observed that carefully in his greenhouse that suddenly some tumors that he had introduced with what's called an octopene plasmid had the aspect from what is called the one of an nopalin plasmid, that is a kind of teratoma plasmid, it's called, because it's all kind of cell types and roots come out instead of a smooth tumor. And he said, but I never inoculated that. How can it be? And he said, it must be that there is this other bacteria in the soil that I splashed on the wound when I was watering my plants. So probably there is conjugation there. And then he proved that if you wound a plant, mix the donor and the acceptor strain in the wound, and you wait 50 days, you have to be patient, then you can isolate bacteria that conjugated. So we took over immediately that method, so then we could definitively prove that the TI plasmid not only exists but is responsible for that.
But that was a radical idea. I mean, it's still a radical idea to this day that a bacteria can conjugate with the eukaryotic cell, right?
Oh yeah, sure. So
Profound it was?
no, absolutely. Well, a lot of people laughed with us because you have exactly the same type of tumors if you do crosses between Nicotiana langsdorffia and Nicotiana glauca, I think. There is what, and that was called genetic tumors, and there is no agrobacterium around, and the aspect is exactly the same. I don't know if meanwhile somebody has taken it up to really analyze that, but that was for all plant physiologists sufficient to think all these Belgians, we shouldn't consider them. So luckily we were invited at Gordon conferences, and we could talk about the story, and Larry Bogorat was interested, and a lot of other people were interested. So it was accepted that it really was, that was the story. But we had to prove it, that it was DNA transfer. And in those days we were already was good friends with Rich Roberts. Rich Roberts, Nobel Prize pricing and so on. He was in Cold Spring Harbor working on restriction enzymes. And we had very early access to restriction enzymes because it was important for the work that Walter Fiers did with SV40. So we used it for mapping the TI plasmid and for trying to make a physical map of the plasmid, because these large plasmids you needed to have a whole set of enzymes. There was Southern blot didn't exist in those days, so we cloned all the fragments and then see if they would overlap. And well, that's a long story, but the most is that we had access to restriction enzymes, and actually we made them a lot themselves on instructions of Rich Roberts. And then the winter, I think, of 74, 75, I was 2 months in Cold Spring Harbor for making more enzymes and learning techniques. And in those days, the first manuscript came in from Ed Southern on the Southern blotting. I think it's only published in 77 or 78, but the first draft came in, I think, around 75. I should look up the exact dates of so. So I saw that this Southern blot, that would be the method to go. Maridel in those days tried to do cut curves to show homology with cloned fragments, and there was an enormous work. And Jeff always had the good philosophy: if it's too much work, think harder, find another way, but don't waste your time by doing the work. He said he learned that in the UK, that if the method was too long, go back, drink tea, think better, and then it will you can do it with less work.
So what was the chemistry between you and Jeff that made the environment so special?
I don't know. We know each other as students. We were in the same political, philosophical organization that we call it the Humanistic Association. That was a kind of lay movement of humanism and taking care for society. As a biologist, he was very under the influence of the declarations of the Club of Rome, how bad the planet is going. So I also was a political militant, so we always worked close. And so when he became full professor, Fiers and I, we had already formed a kind of lecture club. The three of us at noon, we met and discussed literature. That was in the 60s. So then when he came over, we sheltered him. We organized the labs and we fused the lab. So we just did things together. I went on a little bit with Fiers on RNA phages, the proteins, because as you cannot sequence, you have to sequence the proteins. So I started then a protein sequencing lab, and at that way I could then feed the orders of the oligonucleotides that they had, and we were sure that the sequence of RNA. So that went in parallel, but with Jeff we focused on agrobacterium. And then, for all our life we were further always together till in 78 he became then director in the Max Planck. So he started in the Max Planck, and because he wanted to stay also in Europe. We both had offers. Laurie wanted both of us in Harvard, and we both decided that we would stay in Europe. We were the winter 75 restriction enzymes. So when I was back, I asked a technician to start doing that, but of course I should have been wiser and write to Ed Southern or asking Ed Southern. But we were very timid persons. We considered ourselves as dwarfs in this big world of molecular biology, and so we did it ourselves till really this technician was able to do it in 78. But at those days, Maridel also had done it, because she also had sent Daniel Siaki to Rich Roberts' lab to use restriction enzymes, restriction fragments, and then do the Southern blotting. I remember that Jeff Strathern was very enthusiastic, but it was never written up. And for a symposium number, a symposium book, it was in the same year it was published in a meeting in Angers. But that was about at the same time. And then we started seeing if there is really DNA transferred, which DNA is it? And then that's what we call now the TDNA. And then with this mapping that we did of the fragments, we said it's just one stretch that is transferred. And I still remember the sentence of Maridel: nonsense. I don't believe it. The week afterwards, she called, "You are right. My manuscript is ready. When do you publish? That was just 1 week or maybe 2 weeks, because in those days there was no internet. No, we couldn't send mails, so it was written either by telephone or written or so. But she was very good sport, so she waited till we wrote it up, and then it was published back to back in Nature, so that there was something like a TDNA. And then later on we found out that the DNA that she showed that was transferred was from an octopene plasmid, and that's a special DNA, never know what it encodes, but it's nothing to see with TDNA. In that plasmid there is another fragment that is was present in this special line as multiple copy. And it gives maybe, well, nobody analyzed the natural tDNAs, what they really are doing, what is happening. I'm always complaining to young people that they are not interested in that surely there must be something important there. But that's again another story.
What year was that?
Oh, that was 78, I think. 78 or 79 maybe, yeah.
Okay,
I didn't look it up, but there are two.
By 1983,
yeah.
4 papers came out with the disarmed vectors that allowed.
Yeah. So that was then a new, really new gene that was in, and that was then the kanamycin gene. That was the first. That was Miami Winter Symposium. Well, it will be exactly 30 years next year. In 2013, so have some festivities for it.
Well, that's great.
January.
I would say 1983 was a great year for plant biology because of those 4 papers that came out where you could transform plants.
Yeah.
And there was a maize transposons that just came
Yeah,
out.
that also finally people believed Barbara McClintock. People analyzed that plant genomes were not something stable, so the all eugenetics from the years 20 could disappear because everybody believed in pure races. So at least we saw that all what she had observed, what was the molecular base of it. No, that was very important.
She won the Nobel Prize that year.
Yes, absolutely, very correctly.
Plant biology had cachet, but it seems to have lost it.
Yeah, well, work harder, gentlemen and ladies.
I want to ask you about the current funding climate, and especially in the U.S., but I think it might be true for you guys as well, that science that is translational is much more rewarded than hypothesis-driven science or discovery-based sciences. And I was just wondering, what role do you think discovery or basic research plays in the big picture of biomedical sciences?
It's crucial. All the innovations come from there. If you think about the plant science, all what we want to know further on hybrid vigor, how so many items that will be important. All the epigenetic phenomenon that then later on fixes as a mutation, that are the basis of evolution, and in that way understand how you have to adapt when you have stress, because everybody wants drought-resistant plants, but there is such a knowledge that we are still missing. So all this we know already at the moment that it will be big complexes of proteins that will do the regulations. We have isolated quite some already, but many more will have to be done and find out by comparing what happens in nature and see how can you mimic in the laboratory and then finally understand. No, it's always everything comes from fundamental research, but the problem is that the translational research is blocked worldwide. People don't realize it because you see American companies that are successful like Monsanto or DuPont de Nemours, but what they bring to the market is very, very limited compared to what could be done if there was really intensive science and if the applications were not so expensive. All the regulatory, even for Monsanto, claims that it takes them really several hundred, sometimes $1 million for really going from the step from the lab through the whole regulatory process. And don't talk about Europe. Of Europe, there is nothing that goes to the field, so nobody wants to invest in it. So at the moment, we are very lucky that we are supported for fundamental research. All what my successor Dirk in Zee does here in the lab is all fundamental research. He can do maybe some contract research for a company, but that's not the innovation that the society will see. Society appreciates that he does fundamental work, so we are lucky. But that will not go on more and more. So we will be also in the same stress situation that the States also is, that if a small medium company, a startup cannot make new novel plants because it's too expensive and too insecure how the regulators will decide, then we are in problems. And I think it's globally that we have to organize that this story stops or so. And I'm afraid that it is not Rio +20 that will do it in next month of June, where the world comes together to say however all these regulations should work. It is not for progressing with science that they meet there. It's to make things more complicated.
Americans seem to have bought into the genetically modified crops a little bit more than Europe. What are the issues for Europeans about genetic modification of crops?
Well, some people and that are actually very big and powerful organizations like Greenpeace and Friends of the Earth started the rumors that GM is dangerous for health and that GM is surely very bad for environment because all biodiversity will disappear, and anyway that is nobody advantage in the society, that it is only good for the big American multinationals and that we should keep them out of Europe and that Europe should go his own way. And that was about the nonsense that has been said, and that was accepted because really scientists thought it so ridiculous. Why shouldn't do it? Our political leaders will do it, and our civil servants will explain it. It's their task to do politics. Why we are incompetitive scientists, and those on the other side of the Atlantic are very good, so we have no time to lose. We will not do the politics. We believe in our government and in the European organization. They will solve that. But they absolutely didn't, and they became also afraid that it was dangerous. And I think it's only rather recent that scientists in public institutions start to come together and to work on it, to communicate and to explain to society that we have only one planet. If overpopulation does so much damage, we have to solve the problems, and the best to do it is using science and engineering to solve it, and not manifestations or prayers or saying we go back to the good old world, because with the 7 billion we are, it would be a bit difficult. In the States, if you see the Sierra Club and quite some other groups, and especially in California, you have quite some groups who also think that GM is bad for environment. So I always told Nina Fedorov that she shouldn't believe that the Americans are so superior at that level that it needs some education and everybody can use it. If you see how in the United States the attitude towards the concept evolution is, you know that some beliefs can be very disturbing for science. So why not that also?
Yes, it's true. It goes across many levels. So I wanted to ask you a little bit about your unique blend of science and politics, and you're now the head of the Institute of Plant Biotechnology for Developing Countries. Maybe you can explain to us what the mission of your institute is.
Okay, well, the idea started already 10 years ago, but it was a bit difficult to build it up and to find funding, because I fully agreed. First priority is have solid fundamental research, and that is what my former lab, that's now called Plant Systems Biology, is doing. But already in the 90s, because at the end of my career, I knew that state support to science, if it's democratic, it should make distinctions, not too many distinctions, and should be rather distributive, and everybody should have opportunity. So if we wanted a big chunk for really that we claim that we were doing something important, find your way to do it. So luckily at the end I could convince the regional government, what we call the Flemish government, to set a special institute for biotechnology. Of course, I thought it would be plant biotechnology. The minister in question was a Christian Democrat closer to the Catholic University, so was very well known in medical field. So most of it was the medical field, but plants were a really substantial part from it, because that was in the 90s. There was still a divide between state universities and free universities in the Flemish region. So that was, but luckily now it's through this Institute of Biotechnology. It was a virtual institute. So there is an office, and it is at the universities, some labs that start to share knowledge, share technologies, and work together. Well, we didn't have too much to share because we were the only plant group, but okay, it worked, and then the group developed. But I already saw then that, well, for applications, life became more and more difficult in the 21st century. So that there was needed that the applications also should work for developing countries, because there is the highest need. If you see 1 billion and 100 million people who are either starving or at least malnutrition conditions. So at that moment, we have to see, can science stimulate better governance? Because it's, of course, better conditions for living and for income that they need, but can we help with science that they would have a better income and influence, that authorities would have an interest if they suddenly see it doesn't have to be the traditional. What can we do? So that was the early steps, and later on we saw that also in Europe, with all the action against GMOs that overall Europe, these methods were not progressing very well. So we think that the task of IPBO will be really plant biotechnology outreach to a larger group of people than normally the multinational companies do, because at the moment the applications are 6 major companies who do it. So Dow, DuPont de Nemours, Monsanto in the States, BASF, Bayer here, and Syngenta, 3 here and 3 there, although most of them still do their work in the United States. Now BASF, Syngenta are very active in North Carolina and so on. And so try to do the outreach here and see that in that way, by example, the mildew-resistant potatoes, so the Phytophthora infection, people understand immediately that it is an advantage. At the same time, if we can help for the bananas in Africa or for the Jatropha, that are a kind of crops that people can understand, that and the technology and the knowledge exist, can it be done? And so now we are very happy that Brazil is progressing very well with their own GM constructs, and that they recently what they did with beans, it's a million farmers that will have an advantage of that. So we see that by an increasing our cooperation with Brazilian universities and this stimulating the application of the science together, that we will be in the good direction. So that is the what we consider as our mission. See that this fundamental science sees that it can be applied, so that they can be subsidized, and that they can do still go on in doing this competitive high-level fundamental research.
In the course of your career, you must have trained hundreds of students and postdocs from all over the world. I know they came to your lab. And so I was wondering, what is your advice to a young scientist today in 2012?
Well, it's the same as always. Enjoy what you're doing. See that it is important. Talk to people. Ask questions. Never believe anybody, and ask questions again and again. Don't be too pushy, but know that you have to go on, and mostly learn to work with your co-workers and really find a group that have a good spirit, and then the whole group will advantage, because humans are social animals, and we sometimes ignore it, that we are like that. Our biology is like that, our neurobiology is like that, and in that way we will use the best of our neurobiology to find the solutions of the problems.
Okay, well, thank you, Marc.
Please.
I want to thank you for leading such an inspiring life.
Well,
It's been a real pleasure
see,
to have this conversation.
I didn't stop yet, so but.